the second lecture of the the two in vivo imaging lectures. So this was initially supposed to be a hands-on class. Of course, that this format that we have right now does not lend itself very well for hands-on classes. And there's already other people who have, initially the plan was to go a little bit over the calcium imaging analysis, and we're going to do that. I'm going to recommend a bunch of other resources for you guys to follow if you're interested in a webinar kind of experience where you follow a particular pipeline for analyzing data. But here we're going to cover things a little bit at a more general level. So not covering one particular tool, but covering what's around. And before we do that, I'll incorporate a little bit of feedback that I got from you guys of things that were not exactly covered last time, and which are really, really important. So I see there's something happening. Oops. Do we have, okay. I thought someone was raising their hands or something. The first thing is filters. So we're going to cover a little bit about so in order to image, we generally use fluorescent labels. So fluorophores, Luke has covered this, but it's really important to know how to manage spectra. This is very important basic information that you use on an everyday basis. And, now one point that I should mention is that you can actually image activity without labels, but all of that is very experimental, very low signal to noise. But there have been people who have looked at, for example, map pH and looked at fluorescent lifetimes, which is another form of imaging. To look at activity without labels, but we're not covering any of that here. So with the way that we measure activity in all the things that we're covering today and in the last session is with fluorescent labels, such as a GCamp, for example. And all of these approaches require management of spectra. You have GCamp, which is green, and usually you have a co-label, or very often you have a co-label. And this applies both to fiber photometry, to regular epifluorescence microscopy, to miniscopes. You have filters, you have an excitation light to excite your fluorophore. You have a dichroic and emission filters, very often also an excitation filter that you pump the excitation line into your sample, and then you get a signal back, but you need to filter out your excitation light. So you do not want to have your excitation light in your image, right? And this is pretty much the same principle. So emission filter, dichroic beam splitter, this you'll notice is usually at 45-degree incidence. Whereas the other emission and excitation filters, they are at 90-degree incidence to the sample. So you have your light source, your sample, dichroic, reflects the light into your sample, and then excitation light gets reflected, it doesn't go through, and your emission light goes through the dichroic. So same principle here, it's a scheme of a miniscope, exactly the same thing. Here's the dichroic, here's the two mirrors. Now, you don't always need an excitation filter. So it depends a lot on what kind of light source you're using. And this is important because, sorry, I just need to move my windows around a little bit. Because for some types of excitation, you get, for example, you use a laser that is a single wavelength. You don't need any sort of excitation filter because you already only have one single wavelength there. And this applies very often to LEDs too. And pulse lasers, you usually don't need an excitation filter. However, if you use a different type of excitation, for example, before, and a lot of you are still familiar with mercury lamps. Mercury lamps are white and they incorporate a lot of wavelengths, and that's why you need an excitation filter. Now, I've plotted here using, there's several tools for doing this, but I've plotted the emission and excitation and emission spectra of two fluorophores. In this case, it's GFP and TD tomato, yeah, absorption and emission. This is the absorption peak. We're talking about the curve in the background, and this is the emission peak for GFP, and this is the same for tomato. And I plotted this on a tool from Semrock. They manufacture filters. And then there's other tools available out there. They already have, in this tool, you can choose your fluorophores. I hope you can see my cursor, my pointer. You see your filters that you can, from their catalog, that you can use, and light sources and detectors and so forth. So I'm going to spend a little bit of time going through this. First of all is you have your emission. So the GFP, excitation and emission, TD tomato, excitation and emission. Now here, this blue line is your excitation laser, which in this case, I chose a 473 laser line. It's a continuous wave laser. So it's a single line and it excites GFP. Very often you can't excite your fluorophore right at the top of its absorption. And this is fine. And the reason why you can't do that is because sometimes you're also exciting more of your other fluorophore that you have in there, or sometimes it's just that the best laser line that you have, the most powerful one, it doesn't completely coincide with the maxima here. GFP, in fact, is optimized for 488 lasers, which is a laser type that used to be very common before. Now we have different laser lines. So it doesn't really matter if you're not exactly at the maximum. So one thing to keep in mind is that if you have an excitation maximum and you excite at different positions, for example, if you excite at E, D, C, B, A, you're not actually changing the emission wavelength of the light. You still have the same curve of emission. So if you excite a 450 or if you excite at 500, your emission light is not going to be a different color. What changes is how much emission you'll have, because if you excite at 450, you're much less efficient at exciting this fluorophore than if you excite, for example, at 550 or at 500. So illumination at a higher or lower wavelength only affects the intensity of the emitted light, and this is an important thing to keep in mind. Now, so you have your excitation light for GFP, and then I'm using another laser here that I've simulated, and this is a little bit of a fainter line that's right here at 552. So it's right at the 550 line, in the vicinity of the 550 mark here. And this is a green laser that excites TD tomato.
Hey, Tiago, can I ask a question? In the previous slide, so some frequencies suboptimally cause emissions, right? So does that also mean that you can reduce the photo bleaching effect of fluorophores by doing that?
Well, yes, you can, but that is kind of that's not really what you want to do, because you want to see it, right? So if you're saturating, you should always...
Instead of using A, if I used B here, would I be better
No,
off?
what you want to do is really use A and use less intensity of A if you're photo bleaching. So there's one point, if you're saturating your sensor, if you're saturating your imaging system, you just don't want to use the same intensity, so you reduce the intensity. Because using B or C will not really reduce, yes, it will reduce your photo bleaching, but you can't see what you're doing, right? You can't see so it kind of defeats the purpose, because your main purpose is to see your fluorophore. And then you worry about photo bleaching. In fact, it's a problem because you're going to be pumping more laser than what you need in order to excite your fluorophore, right? If you use B or C.
Oh, yes, got it. Thanks.
Okay, so then we have this other laser light, and you see this one is almost at the very peak of TD tomato absorption. Now, we shine this light into our sample, and we get emission light from the sample back, which is going to be these two peaks. So this is the GFP emission light. This is the TD tomato emission light. And the first thing we need to do is to get this dichroic mirror here. The dichroic mirror will separate your excitation light, which is the laser lines, from your sample here, from your emission light from the sample. And what you see here is I'm using this dark green light is your, and I just noticed a problem here, but this will become a trick question. So this dichroic is this LP02561RE. What does this mean? So LP means it's a long pass filter. It means that anything that's longer than 561 will go through, will go straight through. There will be transmission. And this is what you're seeing here. You see 561 and it goes straight through. And anything lower, so it's a low, it's a low pass filter. And low pass is actually for frequency, not wavelength. And this here is absolutely or nearly zero transmission. One thing to keep in mind is that if you're really, really far from the area where the filter was designed, you start, say here in the 400s, you have these weird effects where you can have some sort of transmission. So it doesn't block everything throughout the whole spectrum. Very often you have these effects here where you're very far from the region where the filter was designed to be used. So this is something to keep in mind frequently. So what you're having here is that you block the excitation lights, but you allow the emission light through. Now, what's wrong with this choice of dichroic here? Anyone tell me? So you have GFP, TD tomato, emission. You have a dichroic here. You want your emission light to go through. What's not going through? So will you get, will your GFP emission reach your detector?
No, it won't.
It won't go through.
Right. So you get blocked. You'll get
right.
So this is an example of a poor choice of filters, and it was actually accidental. But what you do in these cases is you have filters that have notches. So you can have filters that have notches to reflect the two laser lines specifically. So how would a filter like this look like? So it would be a filter that has, for example, transmission in all of this area, say from 500 onwards, but which blocks anything below 500, and then has a little area here that it blocks the 562 laser line. So that would be the most appropriate filter to use here if you wanted to look at both of these channels simultaneously. However, if you've ever used a regular epifluorescence microscope, then this is what you do. You have different filter cubes. So this set of filters, so the emission, excitation, and dichroic, they're usually contained within a cube. And you can change. You go from the GFP channel, you turn the filter cube, and you go into the other channel, right? Now, just a dichroic is not enough. You still need an emission filter, especially because the dichroic doesn't give you 100% reflection. So it transmits a little bit of the excitation light. And remember, your excitation light is usually a lot stronger. It's a laser. It's a lot stronger than your emission light from your fluorophore. So you have these emission filters. And here the emission filters are this green, the light green, and the bright red here. So these are the 514/30 and 582/15. Do you guys know what this means? What does this mean, 514/30? So
it's the like
Yeah,
slash 30 is on either end of whatever value that is.
Yeah. So it's actually 514 is the center, and 15 it's 30 width. So it's 15 to each. So it's split between the center. So it's 15 nanometers to each side of 514. So that's why you get the filter to the transmission to go up just before here, just around 500, a little bit before 500, and then it goes down again at 545, or actually 530. Sorry, 530. Yeah. So you get this notch. So this is what this means. 514.30 means it's a notch that is centered around 514, and you have 15 nanometers on each side where you have transmission. Okay, so now there's a couple of problems with this plot still. One of them is, do you think this can be misleading? What do you think can be misleading here? For example, why are the peaks of the emission of the fluorophores here both 100? What are they normalized to? So what's happening here is that the peaks of the fluorophores are normalized to, each fluorophore is normalized to itself. So this is the absolute, it's normalized to the maximum of that peak. So this doesn't tell you whether TD tomato is brighter than GFP or vice versa. And this can be a problem. Why can this be a problem? Well, because what happens when you have a lot more of one fluorophore than the other, right? Say if you have a fluorophore that is much, much brighter, and you even have more of that, what happens is that you have bleed through into the other channel. So this is something to keep in mind. So you always need to check whether you're seeing fluorophore. For example, one clear indication is that if you start seeing a lot of features that are the same in one image, the red image and the green image, that's usually a cause for alarm. And you run into this all the time. For example, we have a situation in the lab where we have a very faint staining for a molecule that's called DCX, and then GFAP, the astrocytic marker. Now what happens is that GFAP is ubiquitous and it's everywhere, and it basically overwhelms the signal from the DCX, which so this is I think what's important to know about fluorescence spectra. So it's a little primer. Do you guys have any questions? Please feel free to interrupt if you have any questions about this.
Sorry.
So the next thing, yeah.
Sorry, I kind of missed, I think I had you cut out for like a minute there. Could you say like the last little bit from the last slide, if you wouldn't mind? After you're talking about the GFAP, I couldn't hear you for like a little bit.
Oh, so what I meant is that you need to be really careful to make sure that even if you separate the spectrum, you have these tails here of emission and excitation. Especially the tails of emission are the most important. For example, if you have it, if your GFP is a lot brighter than TD tomato, which it actually is, and if you have a lot more GFP than TD tomato in your sample, so even though you have this tiny shoulder here of the emission, this could actually be a signal just as strong as the signal from your TD tomato, if you have a lot more of it, right? Because this is percentages, it's not absolute values. So it's something to keep in mind. One fluorophore can overwhelm the other. So was that clear?
Yeah, thank you.
Okay, so one thing that you guys will probably need to deal with is actually assembling these filters, or maybe you have a microscope in the lab and you want to take a filter from there and use it and repurpose it, or build your own filter to combine the fluorophores that you're using. So there's a few rules for this. Obviously, don't put your fingerprints on the filters. Treat these optics with care and everything. But one very basic piece of advice is Filters work either way. They don't have, they work both if you have it turn one direction or the other direction, but they have a preferred direction. And a preferred direction is usually indicated by this arrow on the side. And sometimes it's a logo, sometimes it's a little line, sometimes it's a little dot. And the arrow means whatever surface the arrow is pointing to is a surface with a coating. Now, which side should face your light source? Does anyone know?
The side without coating?
The side without coating, that is correct. So the arrow should point away from the light source. And this is kind of very useful, very practical information. So you usually, and actually very often, you have an anti-reflective coating on one side, and then the filter coating on the other side, and the arrow should always point away from the light source. So the other thing that I'm going to be talking about is two-photon absorption, and how this can pose a few problems when you're dealing with fluorescence spectra. One problem is with two-photon absorption is that if you have a fluorophore that works really, really well in single photon, in your regular microscope, you go to the epifluorescence, you bring your slides, it's great. That doesn't mean that it's going to be a great two-photon dye. So the two things are different. Usually, yes, but not always. So some fluorophores are great in single photon, but terrible at two-photon excitation. And the other thing you should keep in mind is two-photon excitation is usually broader, right? Because I told you last time that you don't have a single line for exciting with two-photon, but you actually have a distribution of wavelengths. So this is something to keep in mind. So, so there's a few resources where you can actually look at which probes are good for two-photon excitation or not. I particularly like this paper by Daniel Fiol, and he has a website to go with it where you can look up, for example, here, GCamp6F. There's a few references, and you can say, well, this is the wavelength that it's kind of a consensus wavelength, 940. For example, for JR Gecko, 1050 to 1150. And there's always a reference, and the paper actually has more scientific, it has spectra and cross absorption cross section, two-photon absorption cross section estimates. The two-photon absorption cross section is a measure of how good each fluorophore is at absorbing two-photon excitation, at being excited by two-photon excitation. Now, finally, about optics and filters. A lot of people are very nervous about cleaning optics, and for a reason. You should be very, very careful. So in general, in the lab, whenever you need to clean any optics, any filters, any lenses, the first thing to use is air, is duster. Make sure they're in a clean place, and use this duster canister. If absolutely needed, you can clean it with a swab, a cotton swab. And there's different things that the manufacturers recommend. Usually it's methanol or acetone or a combination of both. And you clean your optics, and you should always consult the manufacturer before, because different optics can have sensitivity. The coatings can have sensitivity to different substances. And you clean it in an outward motion. So you always remove, you never bring the dust or the dirt from one area to the other. So you constantly, you never swipe again with your dirty swab the same area that you've already swiped. So you use the swab only once, and it's usually in an outward motion. But this is only really if you ever need to. So the other thing that it's worth mentioning is chromatic aberration. So simple lenses do not focus all wavelengths at the same point. So they will bend light differently depending on the wavelength. And for that, you have these achromatic lenses, which are lenses that have, They're composite lenses that are usually made of more that, so this one will be a doublet, so it's effectively 2 lenses. And they kind of compensate for this effect, and they approximately reduce the spread in the focal point that you see with simple lenses. Now, generally, most modern objectives and commercial imaging systems are corrected for chromatic aberrations, meaning if you go to the confocal and you look at colocalization between 2 proteins, and one of them is labeled in red and in green, the other one is labeled in green, the confocal software and the confocal hardware are calibrated so that you see the red and the green exactly the same, that you know that they're exactly the same place and they're not one above the other. However, this is not true for all systems. And here I have just an example from a recent paper from the Diceroth lab, where they're exciting in the mouse brain, and I think this is from orbital frontal cortex, and they're imaging using grin lenses. And grin lenses are not corrected for chromatic aberrations, which means that when you use the red or the green channel, you'll actually have a shift. You will not be, you have an axial shift. You will not be exciting both. So your green plane and your red plane will be slightly off. And sometimes the slightly off can be 5 microns, sometimes it can be 10 microns. In any case, it's off, and it can lead to problems. So whenever you need to do this in a more advanced system, the system that maybe you've assembled yourself, then you need to correct for this. So you put laser. So here you see that one of the laser systems has extra optics to kind of compensate for this. In this case, what these guys are doing, they're exciting. They're using this 1060 nanometers to stimulate, to excite channel rhodopsins. And they want to do this in the same plane because they want to see, okay, this is the cell that I'm seeing. I want to excite that cell. But if you don't have this, you'll end up by exciting a different cell that is either above or below it. So is that clear?
Question about that, Tiago. For things like dual wavelength miniscopes, is that a problem?
Yes, and it's funny that you asked that, because I was going to include something about it. Absolutely is a problem. In fact, there is no widely available dual wavelength miniscope right now. The first ones that have been, I think, widely available are the miniscope, the UCLA miniscope. They're still fine-tuning this, but it took a while. Even though miniscopes have been around for a very long time, there's very few, if any, I don't know if any, that uses more than 1 wavelength. The UCLA is one of them. There's another group at BU that has a dual wavelength. And part of that is they actually solved this problem by, there's a variety of different issues. This is one of the main issues. And they actually do, they separate the 2 channels in time. So they have, because then there you have an extra problem, which is you need 2 LEDs. 1 LED to excite, for example, the green, and 1 LED to excite the red. And you need to have the 2 LEDs have the same light path. So it's a kind of a complex problem. I won't go into it, but those are not commonly used yet. I think they'll start to be commonly used now. So
I think Claire's raising her hand.
Sorry?
Claire is raising her hand.
Oh, yeah.
I was just going to ask, are there types of imaging that, I guess I don't see the chromatic aberration thing as being like a huge, I mean, like it is a problem for certain things, but you could also use it to your advantage if you have like the different wavelengths across depths. Is there types of imaging that, you know, like if you're doing like a Z stack, if you're like separating it by colors, you could excite different fluorophores with the separated bands. Is there any type of imaging?
Yeah, there's actually, it's funny that you mentioned that. There's some techniques that work a little bit like that. So they encode time or space based on wavelength and make use of this sort of distortions. I don't think what you're describing exactly in that format would be very practical, because if you think about it, your at least your efficiency of excitation could vary dramatically between one Z plane and the other, because you'd be having another exciting effectively with another color. And also these differences are very small. So we're talking about, you know, maybe 10 microns. It's enough to be exciting the cell below the one that you think you're exciting. But in the case of the previous example that we just saw, but it's not something that you do a Z-stack with. Okay, so last time I talked to you a little bit about the differences between image forming detectors like CCDs and cameras effectively, and single pixel detectors like PMTs. And I told you that image forming detectors are usually used depths that are less than the mean free path of the emitted photons, because you get this effect with the scattering. So your photons are coming out of out of the neuron that you're trying to study in your brain, and they start boom hitting electron clouds in the tissue around them, and they end up somewhere completely different from where you would expect them. But this isn't always true, and in fact, CCDs have made a resurgence for imaging fixed samples, for example, in miniscopes, which use not CCDs, but a similar light sensor called CMOS. And also, for example, for these broad, whole big window, whole brain imaging, for example, this is from last session, I showed you this example of Voltron. So these guys use CCDs, and part of the reason why they use CCDs is because here they're imaging using a scientific CMOS camera, and they collect images at 3,858 hertz. Now, granted, this is for a very small field of view, a very small portion of it's not for the whole brain, but the kind of imaging speeds that you can get here, which are the kind of imaging speeds that you need to look at action potentials, which are very fast 1 millisecond, 2 millisecond events. These are more easily possible with camera detectors than with scanning methods. So there is a place for using CCDs in cameras in general, even in situations where you're imaging in vivo through fixed tissue. Now, I'll just say a few words about image sensors. So there's fundamentally two different types of image sensors, the CCDs and CMOS. Before, the only thing that you'd use in science were CCDs, charge-coupled devices. And fundamentally, both of them work in the same way. They work with photoelectric effect or variation thereof. They're silicon, so they're basically chips, computer chips. You hit them with photons, and you create free electrodes. So you create a charge, and then you read this charge out of the chip. And the difference is from CMOS and CCDs. So CMOS are now pretty much the sensors that you have in your cell phones, the sensors in most cameras, and most consumer electronics. And CMOS have a different sort of construction. So basically, each pixel on a CMOS has its own attached little amplifier and digitizer. So you've covered this stuff in Luke's lecture, right? So you have this amplifier and digitizer. So each pixel is effectively its own little analog to digital converter, and it has its own little amplifier. Whereas in a CCD, you basically move charge around to one or two readout ports, where you move the charge between pixels, and then effectively you amplify it and you read it out and you digitize it out of a single readout space. Now, what is the problem? Can you guys think of what could be the problem with one and the other approach?
Can you saturate one but not the other?
You can actually saturate both. You know, you usually expose, and I'm going to go into that. You expose your CCD, your image, for a certain amount of time, and then you read it out. And if that is more than the size of your bucket, if you imagine there's the bucket where you collect its charge, then you're going to be saturating. And there's a certain amount of charge that these pixels can hold. Now, I was thinking more on the line of, is this going to be, each pixel here is going to have its own electronics attached to it for readout. That means that you probably won't have the same kind of consistency that you have here in the CCD. And this is effectively a big issue for a very long time. And that's why CCDs were favored for scientific applications. The other problem is that this electronic is usually on top for front illuminated sensors. They work a little bit like our eyes, like in the retina, where in our retinas, our sensors are actually below a lot of wiring, right? Your light goes through a lot of stuff to get to the actual rods and cones, to the active part. And in these cameras, it's a little bit the same. So it's in front illuminated cameras. So this readout mechanism is actually the electronics. They're on top of the pixel. So they actually reduce the active area of the pixel that you use for capturing photons. And that's extremely important because you have less of an area where you're capturing. You want to capture as many photons as possible. And you have this readout electronics on top. Now, there's approaches now to reduce this. For example, one of them is what's called back illuminated sensors that effectively have the active area facing your light source. They're much harder to build, but they have a much better efficiency at capturing photons. The other thing is that CCDs are much slower because your readout is much harder. You know, whereas here you're basically capturing the information and digitizing in parallel from each one of the pixels. In a CCD, you need to move charge around and you only have one output. So this is a much, much slower process.
Can you explain again, what do you mean by you're moving charge from pixel to pixel in the CCD?
Yeah, so you create, this is a relatively minor point, but so it's not something that is important that you'll need to know for on an everyday basis, but basically in a CCD, because you only have one readout, you basically need to shift whatever charge you've accumulated here from your exposure. So let's say you have 10 photoelectrons here in this bucket, in this pixel. You need to move these 10 photoelectrons in a line, and they're all like, all this charge is kind of an Indian file to be read out at the same amplifier and the same analog to digital converter. And that's what I mean. And then there's different ways. For example, some CCDs, they even have, in an attempt to go faster, they have half the CCD illuminated and half of it dark. And the dark half is just meant to hold charge. And while the readout process takes place, while you're already exposing the CCD for the next image. Did that make sense?
Yeah, well, would the noise for CMOS be more than CCD in this situation then?
Yeah, so this is traditionally the noise for CMOS. It depends a lot. So it does, traditionally the noise for CMOS was higher. There was more variability between pixels. Right now, the fact of the matter is CMOS and CCDs don't necessarily have much differences in terms of noise. So that's kind of the summary. There's a lot of things that go into it. I'm going to talk a little bit about some of those.
Okay, yeah.
So how can we, you mentioned noise, and that's one of the things I wanted to talk about. So one of the things that you can do to reduce noise is to basically average different frames. Or sometimes if you're doing the scanning, laser scanning with a PMT, this applies to that too. You can actually average different lines. You can run the same line 2-3 times over again. And what you do is, it's like making a measurement 3 times of the same thing. So that's what so you're basically increasing your N, and you average everything and you reduce your noise. So what you can do is actually average different frames. And this is a very simple way of reducing your noise. Now, the loss is, of course, speed, right? Because you need to take 4 or 5 images, and then you average those. So the other thing is that any non-spatially uniform noise doesn't go away by averaging. For example, if one area of your chip has a different response, this is kind of relatively rare. But in general, averaging is a very very often used technique, very common technique to reduce noise. So you repeat your measurement a few times, you average it, and you average out the noise, because the noise is varying temporarily. So you will not have the same amount of noise in one measurement and the second one. So you average all of that out. Does that make sense? So another way to reduce noise, and this is kind of specific to CCDs, is to do binning. So with binning, what you're doing is you're basically saying, well, this is the standard CCD, but I'm going to bin all the charge from a 2x2 square. So you reduce you actually sacrifice your spatial resolution. You put all of these electrons together, and you read them out together. And then say, if you do a 2x2 binning, you'll have 40 electrons being read out for each pixel, for each bin pixel. And let's say if you have a read noise, a fixed readout noise in your readout system that is 10 photoelectrons, so you always have that 10 photoelectron fluctuation. Here, by binning your charge, what happened is that you're actually going well above the noise in comparison to the situation where your signal to noise is about 1, because you have 10 electrons, you're reading out 10 photoelectrons, but your readout noise is about 10 photoelectrons. So by binning, by increasing the number, increasing your signal and making it go above the noise, by binning is a good way without sacrificing. So here you sacrifice spatial resolution, but you don't sacrifice time. So you're still going as fast as you're still going fast, but you're now, in fact, you're even going faster because you're reading more electrons at more pixels at the same time simultaneously. But you sacrifice your spatial resolution. So one area where CCDs are still relatively more often used than CMOS chips is for whenever you have very, very low light applications. You know, this is kind of the equivalent. One area where you guys have probably seen this is these military light sight applications where these guys wear these cameras on these goggles with super light sensitive cameras that allow people to see in complete darkness. So these are, there are different technologies for doing this, but the most common technology now is these CCDs that have what's called a multiplication register. So they undergo in a process here that's called impact ionization that multiplies the photoelectrons before the readout. So you get a lot more photoelectrons out from a single pixel before you actually read it. So, and you go through this stage of amplification. It's a little bit like a PMT, the analog of that would be a little bit like a PMT, but this is in the output of your CCD. But in practice, this also creates some multiplicative noise and shot noise. And now even these EMs, so these are the electron multiplied CCD, electron multiplication CCD, I think that's what it stands for. So even these are now struggling to compete with some of the more sensitive CMOS chips. So now, okay, so this is a problem that's very similar to taking photos with your camera. So you have a camera, you're at the microscope, whether it's in vivo or not in vivo, or whether it's just your camera at a party that you're at on your cell phone. What's the optimal exposure time? How long should you expose your sample for? Now, if you're at a party, the phone decides this for you. But you know that very often, if you're in a really low situation at night, you get your image is not good. A, it's blurry because your exposure time was too long, so the image moved while you were exposing. Or B, you've accumulated a ton of noise, and you get this image where, yeah, you can tell who's there, but it's just a horrible image with a ton of noise. So what do you want to do? What do you want to do? How do you optimize? How, if you're at the microscope, how much should you expose your sensor for, or how much in the analog for laser scanning systems is how long should your dwell time of your line scan? How fast should you scan a line in your system? Now, the best simple answer is to say, well, make the best use of a dynamic range. Now, you guys remember what dynamic range is from your lectures with Luke? For example, an 8-bit imaging system. So how many levels would you have there? What's a mathematical formula for an 8-bit system? How do you calculate how many levels you have? Yeah, Claire? Do you want to say it?
Is it 2 to the 8th or no?
Yeah, exactly. So you have 256 levels, usually define that between 0 to 255. So this is your bit depth. And hopefully that's matched to the dynamic range of your sensor in an imaging system where everything is kind of designed together. And what you want to do is you want to look at a histogram. And I think you covered these with Luke, right? So if you go to image J and you plot an histogram, this gives you the count of pixels. So this is 0. How many pixels are exactly at 0? And how many pixels are at 255? So this will be absolute white. This will be absolute pitch black. And there's these gray levels in between. Now, what you see here is that you have your signal kind of starts kicking in, not quite at 0, but a little bit after 0, and you have very little stuff that is at 255. But what you want to do is really have your brightest pixel be 254, and your darkest pixel be 1, and then you kind of use this to optimal effect, right? Using the whole range of values that he can use. And then you adjust your exposure or dwell time to achieve this. And one easy way to do it is to use these color maps. So this fake color so this is one example. This image is from a gel, but that's the only thing I could find on the internet. But this is your actual image here, an image of a gel bands. And you see there's some very faint gel bands here where my cursor is. And you can use a fake color scheme where your 255 value is red and your 0 value is blue. And what you want to do is to just make sure that you don't have any zeros and you don't have any blue and you don't have any red. So you adjust your exposure to control for that. So did that make sense? So now, the reality is a lot more complex because in fact, your dynamic range of your sensor is not always enough to see all the features that you're interested in. And you need to optimize whatever image you acquire for the features you're interested in. And this is actually from I found this on the internet from this course at Stanford. And this is an example of an image taking with a high dynamic range sensor. And this is an example of the same image using different integration times. So this is 1/16, 1/4, or the full integration time. And you see here that if you get this, if you don't saturate the chair, you don't see this guy at all. And if you saturate the chair, then that's when you can see this guy. So it's very hard to get a situation where you're not saturating anything or you don't have anything at zero. So basically, often you need to compromise. And in general, signal to noise ratio is better with the longer integration, but even that breaks down at some point. So there's a point where the noise processes that, where noise increases with longer integration times, they will eventually take over. But that's usually not the case. So your signal to noise ratio will generally be better the longer integration time is. Okay, so how was that? Was that clear? Now, Luke, is there anything you'd like to add to this, or anything that wasn't? Just saying, but if you think of anything, feel free to interrupt at any point.
Yeah, sorry.
Yeah.
I have a quick question about the previous pictures.
Yeah.
There's one, the top one. You can see the person, but you can't. The chair isn't saturated. What's the difference between the top one?
The difference is the sensor. So this sensor has a higher dynamic range.
Oh, gotcha. Okay.
So that's a thing. But very often your sensor that you're using doesn't have the dynamic range to capture this whole range of, because it may be that in a sample you have something that is very, very bright, and that's not what you're interested in. And it's impossible to get that very, very bright thing to be not saturated, and then your process of interest to be visible within a reasonable use of your dynamic range. I mean, one example is if it's really sunny outside, I have a window behind me and you won't see my face, right? Because you're optimizing your dynamic range to capture the room with a very bright window behind. So that's one practical example of that. Okay, so I have a couple of slides now very quickly about emerging techniques. Some of these are not really emerging at all in the sense that they've been emerging for 5 years or so.
Tiago,
you're still kind of a little cutting edge.
Tiago, I was just going to say it's about halfway through now. If it seems like maybe this is a good point for a break.
I have 3 slides, and then that's it for a break. Just because then we're going to do mostly image analysis. So one emerging technique is this all optical circuit interrogation. So this is gives you the ability to image neuronal activity while optogenetically activating or inhibited selective neurons. And this is kind of the holy grail, right? It's kind of the holy grail of imaging and studying the brain, is that you're imaging, say, this is from a review by Valentina Miliani and a few other people. You have all these neurons here, you're doing a behavior experiment, and now you want to say, well, that neuron, it came on when the mouse did this behavior. So now I'm going to activate this neuron and this neuron and this neuron. I'm going to see what the result is in the circuit. And this is, and being able to do this live and full time and even unsupervised, so that the computer itself will know, okay, whatever neurons become activated at this point, we're going to suppress them. And we are already developing the techniques for this. So there's of course the sensors. So we have the GCAMs and the JR geckos, a bunch of expression techniques for expressing this transgenic mice. And then you can use different imaging techniques. But then for the manipulation, you can use the channel rhodopsins and the different variations thereof. And you have techniques that allow you to use a different channel to selectively excite or inhibit particular neurons. And this is kind of as I said, it's the holy grail of doing this. This is kind of the end game for the whole imaging communities, when we'll be able to do this in large scale and very easily. Switch on, switch off, see what happens. And there's a couple of examples here. So for example, this one is from a paper in the barrel cortex, where you look at transients with sensory stimulation, particular transients of particular cells, and then you can photostimulate and you replicate. You can activate the same cells. Or in this paper here, this is in the hippocampus, and these guys, they simulate what it would be like. So the kind of pattern of neurons that are activated at a certain location, so they can kind of artificially recreate that location by stimulating the cells that are usually active at that location, so with place cells. And on that note, since you guys don't have a lecture next Thursday, if this is something that is of interest to you, there's this, the Optical Society of America is having a lecture by one of the leaders in this field of all optical interrogation of circuits. That's Valentina Emiliani. And I highly, highly recommend her lecture. That's on Thursday at 10 a.m. and it's free to attend. The other emerging technique, and this is really, I'm getting to the break, that I'd like to talk about is light sheet microscopy. Now, this is not really very useful for a mouse because light sheet is based on exciting a sample from the side with a sheet of light. Exactly, that's why it's called light sheet. So whereas in two-photon microscopy, you create only a small point of excitation and you scan that point across a Z level. Here, what you do is you excite a whole, on a single Z plane, you excite a whole Z plane, a whole image simultaneously with a beam that's coming in from the side, and it's a flat beam, perfectly flat. So you see here, this is laser scanning. This is wide field, where you have the light coming in from above. You excite the whole fish. This is a zebrafish embryo. This is a confocal technique. It will be similar for two-photon. You're coming in from above. And this is light sheet fluorescent microscopy. You come in from the side and you detect from above. So this requires side access, which mice don't have because you have your skull here on this side. You can't. But fish are mostly transparent. Then you can do this. And the advantage is you can go extremely fast, and you can actually image a whole brain and a vertebrate animal, which is quite amazing. And you image this with single cells. So you see here that you can see the single GCamp expression cells. So this is a paper by Misha Ahrens. And it's really, truly amazing how you can actually image a whole brain of an animal during a behavior, and you can do behaviors with these embryos of zebrafish. And you have pretty much access to every single cell in the brain of this animal, which is incredible. So yeah, and I think this is the one point where I'd like to stop for a break. Any questions? Okay, so it's 11:31, so we're doing a 5-minute break around. So let's reconvene, say at 11:37 or so. All right.
I mean, one thing, Tiago, people use light sheet for clarified mouse brains.
Yeah,
so
they
it's
do.
Not, in vivo might not be useful, but in practice, probably a lot of people are going to use it.
Right. I forgot to say that. It's very useful for mice, but it's really dead mice that you can image. You cannot do that very easily with living mice.
So there's this guy in Dijsselrath lab. I think it's not published yet, but this guy Aaron Andelman, they have he's telling me at SFM that they have single cell resolution in zebrafish now with light field microscopy.
Oh wow.
So they can image the entire CNS with like 100 hertz, temporal resolution.
In zebrafish.
Yeah, like cellular resolution. It's like whatever 100 hertz.
Yeah, that's quicker than this.
Yeah, right, yeah.
Much quicker, yeah. Okay, so I'm gonna go for some water and I'll be right back.
Hey, Luke. Can you hear me?
Yeah.
You mentioned that we were getting the optical coherence tomography microscope, is it?
Yeah.
What you were saying? When are we getting that again?
So it's all assembled right now, except there's like a couple parts that are missing because they're in China.
Okay.
And so we had them custom manufactured in China, and they're like sitting in a warehouse somewhere. They're finished, but they can't ship them to us.
So once this thing is over.
Yeah, yeah. So it's sitting on a shelf in my lab. So.
Okay, cool.
Okay, I think it's time to start again. I think that most people are probably back. So for the next few slides, I'm going to try to address the question of you have all of this data, so what now? How do you get about getting useful data? How you get about getting meaningful information out of all the images that you collect, because so far I've mostly focused on collecting images. So the first thing I want to say is that whenever you're acquiring imaging information, your best friend is going to be ImageJ. So this is a free open source imaging platform. It's actually made at the NIH, and it's available in a bunch of different distributions, which are basically ImageJ plus a bunch of plugins. And a lot of people contribute with plugins, and it's great for exploring your data. So pretty much whenever you acquire an image, a video, you load it immediately into ImageJ to look at what it looks like. And I recommend the one distribution that I usually use, it's Fiji. There's others, but I recommend this. It comes with a lot of useful tools already, and it's a one single download, and you have a bunch of different tools that can be very useful. So there's a lot of analysis for things that are not time-varying signals, which are usually morphology. Anything that is static, that's relatively simple. You know, there are some. I'll take that back. There is something to it, but it's generally conceptually easier than time-varying signals. And usually you need to do this analysis for example, for spines here. You see here, this is actually a beautiful image of spines. They were imaged over time course of over a year. And humans are much better at detecting a lot of these features than computers. So you can do stuff that is computer assisted, but generally you require human supervision. And there's a lot of tools out there. One of them is commercial, is Neurolucida. There's also this thing called the simple neuronal tracer. So these are good for both spines and axon arbors and dendritic arbors. And there's ImageJ that has a bunch of different other plugins. So simple neuronal tracer is actually a plugin for ImageJ. There's a very good tutorial online on how to use this. And of course you have the commercial systems. There's Imaris too, I think is good for this, a variety of them. And you'll be able to get a lot of information out there on this. So, and that's pretty much all that I'm going to say. So I'm going to point you to these tools, and that's it for that kind of data. Now, time-varying signals is another story. So these are signals such as calcium imaging, anything that changes in time, right? Calcium imaging, any other source of sensor, like for example, a glutamate sensor or something like that. And for this, you have a more complex pipeline for analysis. You want to eventually detect activity and events of some sort. For example, you do a stimulation. You want to see what the response of the neurons are to a stimulation, to an odor, for example. And there's a few, it can get a little bit more intricate to get information out of this, this kind of data. First of all, one of the things that you do is, because you're imaging in vivo, your data will have movement, because the mice are not going to keep still. And most of the time, they're not anesthetized, right? So they'll be running on a treadmill, or they'll be running around with a microscope on their head. You'll have a lot of motion and shaking. And this makes it very hard to extract the signals, because you'll get a bunch of artifacts, motion artifacts in your data. Then the second step after you correct for motion is to identify your features of interest, which can be neurons, spines, axons, so forth. And usually you define what's called regions of interest. So a region of interest is basically a set of pixels that will have your signal, which is the set of pixels that you want to analyze. So this is ROI for short. And then when you get those ROIs, you extract the signals, but you still need to do some processing. Sometimes you need to subtract a background. Sometimes you need to find a baseline, or you need to filter these because they're very noisy signals. And associated with that, there's also a part that is event detection on this type of signals. Now, there are a bunch of different tools out there, and I wanted to spend a little time talking just about a few of them. So these tools are meant to make your life easier. Right now, the days when you would develop your own analysis pipeline are mostly gone because it's just become so complex. And there's people out there who only do this. They only concern themselves with, for example, analyzing, their whole careers are built around analyzing calcium traces. And they do it really well. And they've been developing, and they dedicate their entire time to developing these computational tools. And one example of that is CNMF. We're going to talk a little bit about what CNMF means and what it does. There's other tools that use PCA and ICA, independent component analysis, principal component analysis. There's tools that have GUIs. One of them is CalMAN, which actually is made by the same people that made CNMF. This guy, FTQs. And there's Suite2P, which is a set of code and a GUI that is incorporates a bunch of different tools. So actually you can use CNMF through Suite2P and other tools through Suite2P. And then there's many others. SEMA from the Lausanne lab. There's this easy calcium that's supposed to be like the really low barrier to entry and so forth. So this goes to say, do not reinvent the wheel when you're doing this. It's good to look around, look at what kind of signals you have. For example, is a stimulation with an odor, and you want to look at the response of a neuron to the odorant. Well, look at papers that have done that similar type of work, or look at similar types of signal, and look at what people have used. And there's a lot of tools already out there. Do not reinvent the wheel. The other thing is, be prepared to spend more time actually analyzing your data than it took to acquire it, because that's the way it is. Your analysis will generally take a lot longer than your acquisition. Okay, with that said, we'll go and look at the first point, which is motion correction. So I'm hopefully this video will show up fine. You see that this is a beautiful video of CA1, but every now and then, you see, even though this is very good, very, very stable, there is a little bit of motion every now and then. You see this shakiness just now? So that's an example of very mild motion correction being necessary. This video has been corrected, and it actually had very intense image correction. So the cells are more or less in the same spot, but what you'll notice is on the sides, on the two sides of the image, there will be these black bands. So this is where the image shifted so much that the computer that was aligning it had to basically move the image completely to one side or completely to the other. So this is in XY. So you see here how every now and then there's bands on the edges. That means that the computer had to move that image, that frame, in order to align it with the other frames in the video. I don't know if that showed up very well, but so movement is completely unavoidable in vivo, but it can be minimized, and this is what you should do initially. Whenever you can minimize movement, do it at the source. So make sure that your surgery is stable. Make sure that if you need to implant a grin lens or do any sort of implantation in the brain, make sure that your surgery is not bigger than the whatever you need. Make sure that you're not having things sloshing around in the brain, because everything that can shake will shake. The same is true with head fixation, right? Make sure that you have a stable way of fixing the brain whenever you need to. And generally what you do is you align in an XY plane to a reference image. Now, what will this reference image be? Anyone? What do you use as a reference image for alignment? Yeah, actually the answer is there. But so one of them, this very simple approach would be to say, you start with the first image of your movie and you align the next frame to that first image, then the frame after that to that first image, and the frame after that to that first image, and you align everything to make sure that the pixels match. That's good, but your first image is not necessarily your best. So what you usually want to do is look for your more stable frames. And you can do that. There's one easy way to do that is to manually go through your movie and choose a few frames that are stable. That's fine. If you want to be more scientific about it, you can do a cross correlation between different frames and choose areas with high cross correlation, which would mean there is actually, these are areas where the similarity between the frames is very large.
So just quick question then. So by stable frames, you just mean like
no
whatever the majority of the frames are like matching to. Okay.
Yeah, the most stable area, because of course you want to match it. There may be areas that are not analyzable in your movie, but you want to make sure that you have a kind of a consensus plane, which would be the area with the area that is imaged kind of consistently throughout most of the movie. So that should be your reference in general. Now, in two-photon data, beware, because you will lose cells that go out of focus. If your cell goes, remember, you have a very thin XY plane, or relatively thin, compared to, for example, with epi-illumination with a miniscope, where you're just illuminating everything. So you're not restricting your imaging to a single plane. But if you restrict your image to a single plane, yes, that gives you absolutely fantastic resolution at very small points. You see the cell shapes perfectly and everything. But if that cell goes out of focus, you lost it, right? So that's one thing to keep in mind. So tools for doing registration. In ImageJ, I'm going to mention this. I don't usually use it, but some people do, and you still see papers using it. It's TurboReg for ImageJ. This is something you can do with an easy GUI, and it's kind of self-explanatory, and it has a pretty good documentation, and it's been used for a very long time. It's relatively quick, relatively. What works best for me is Fourier transform registration. So things that do registration in frequency space work best, and they're very, very quick, because if you have, say, 1,000 frames, this alignment can take a long time. There's this particular paper that I'm citing here on this tool from MATLAB DFT registration.m. I think this is actually also used in Norm Core E, or at least as a similar method. And the idea here is that cross-correlation between frames, which is one way that you can see whether the frames are aligned or not, they're really the same as a convolution of a frame with its frame of reference. And convolution is a multiplication in the frequency domain. So what you can do is if you move everything to the frequency domain, the kind of operations that you're making are actually multiplications, and it makes it computationally much easier for the computer, much quicker for the computer to perform this alignment. And that's why these tools have been extremely useful. And there's tools for MATLAB, there's tools for Python, and there's a more complex, the evolution of this is tools such as Norm Core or Norm Core E, I don't know how they pronounce it. But basically these are whole pipelines for aligning, for doing motion correction, for doing registration of movies. And they actually divide the image in different patches. And for each different patch, you do a correction. And then you divide it into smaller patches, and you correct again, and it's like an iterative process. What this allows you to do is it actually applies shifts, as you see here in this bottom image. You can apply shifts to different areas of the image that are going to be different. So here, for example, you see that because the brain is not completely rigid, you can actually have areas that shifted a lot more than others in the same image. And what this tool allows you to do is, in a time-efficient way and computationally efficient way, correct these things. And the results are really amazing compared to other methods. So finally, how do you check if an image is registered? Well, there's different ways of doing. One of them is, of course, cross-correlation. You should see that there is a high cross-correlation. The other way of doing it is that the principal components should be, if you do a principal component analysis in your sample, whatever's varying, your variance should come from your activity, from your cells, and not be from stuff that is outside of your cells. So your main components in your sample of variance, and your main sources of variance should be whatever's should be your cells and the time varying signal within your cells. And SWIT2P actually has some benchmarking. So there's some ways to verify that you're actually aligned and how well you are aligned. And I'm going to recommend also, there's these videos on the internet for some of this stuff. I'm going to recommend a few of them. And there's one by Carson Stringer and Marius Pacicario on SWIT2P. I'm going to get to that. But if you're interested in learning how these things work, there's actually a ton of resources online. So the next step is, okay, you've aligned your movie. So you have a movie with cells that are flashing. Now, you want to detect signals, calcium signals, for example, out of this movie. And the first easiest way, and the way that people have started doing this, is just segment the image into regions of interest. And what you do is basically, you can do it in image J. In fact, you should do it. This is one of the very fundamental things that you should do whenever you're analyzing before you start anything. You have your image. You have the cells here. There's actually, I don't know why. The first thing you should do is actually do a Z, and I thought I messed up here. I thought I had you should project a Z stack. Basically, once you have aligned your image, you project a Z stack of all of your, you have all of your different frames. Say you've imaged for 10 seconds at 5 frames per second. Then you should have 50 frames. And then you average all of these, and you get something like this. Let me see if this works. You see, this is an average image, and it's usually very high. Because it's averaged, you can tell the intricacies of your structure very well. So, for example, you see these cells here. These cells are labeled with JR Gecko, and you even see, because it's an average image, you see this shadow here. You see this? What do you think that is? Why do you have these dark shadows?
They're like blood vessels.
Yeah, exactly. So you see the blood vessels, they cast a shadow over your sample. So, and then if you want to see how a cell, what kind of activity a cell had during this time that you captured the movie, you literally just select ROI, which is what I show here. So you select it on image J. You take, for example, the circle tool. This is a very large, so that there's probably 1,000 cells in here. You select one cell and then you plot it. And there's a way to plot the activity. And you see this cell had this very large event, and then it was kind of fired and then fired again. And then it went silent. And it had this very large sequence of events during the movie only once. So that's the more basic way of analyzing calcium traces. So you can even hand draw, and you can, yeah, Claire?
I have a question about setting Z stacks. So in the example you just gave, what would be like the thickness?
Sorry, I'm calling it a Z stack. This is not a Z stack. This is a T stack.
A T stack.
Temporal.
Oh.
Yeah.
Oh, okay.
Sorry, the thing is that the option in image J actually calls it Z stack.
Yeah.
But it can be whatever stack it is. It's just that other dimension is time here. It's a time varying signal, but you stay in the same plane. Yeah, I'm sorry. This is a little bit misleading. Yeah.
Okay.
So when I show you this is even though the option in image J is called plot z-axis profile, this is not the z-axis that you're plotting, it's actually the temporal axis. So each one of these slices here is a different time point. So what you're seeing here is time domain, and this is fluorescence, raw fluorescence image. So fluorescence counts. So your you can hand draw cells, and people do. It depends on what you have, and it depends on how much investment of time, what part of it. There's nothing wrong with that. People still draw cells manually. Depends on your project, depends on what you need to do, depends on how many you have to draw. Obviously, if you want to draw cells manually in a sample with 1,000 cells, this would be a mess, right? It's just too many. But if you have 5 cells, that may be the way to go, right? I mean, why bother trying to learn a tool that can be very complicated if you can do it like this? And so you start with the average projection of a registered movie, and then you can use a simple thresholding or a watershed algorithm. There's different algorithms. So here, this is actually an example from a pre-GCamp paper. This is taken using a bolus injection of Oregon green bapta, which is a small molecule dye, and you inject it, you have all of these neurons, and these neurons are very nice because the signals are very nice and round. The astrocytes are in yellow because they've been co-labeled with a red stain. And what you do is you basically set a threshold, and you can set an upper and a lower threshold for saying, so you only take areas which are above a certain level that is above the background. And then, you may get areas where there's different cells touching or very close, and these cells will appear, they will appear as being joined together. But then you can apply different filters. For example, one of them is applying for a size because you have an expectation of what the cell size would be. Or the other one is the shape of the cell, things like the eccentricity. Is it going to be like a round thing? Is it going to be an elliptical thing? All of these filters you apply, and you can create code to very easily detect all of the different ROIs in here, so all the different cells. And then this is a mask that you apply to your image, and then you can automatically extract the signals from each one of these. Here, I think it's 113 cells that were detected in this image, right? So this makes your life a lot easier than just drawing things. Now, this approach is good and works well for cells that are relatively round, homogeneously filled. It works great for this OGB stuff, but it'll fail for GCamp because GCamp is much harder because, first of all, your cells are actually donuts, right? There's also a more serious problem with this, is that drawing ROIs, whether you do it by computer or manually, fails when the neurons are spatially overlapping. For example, when you have very dense structures like the dentate gyres, this is an image of the dentate gyres. Can you tell me here where a cell stops and where another cell begins? Because I can't, for me, it's impossible, right? And then you have situations, for example, you have the dendrite of the cell superimposed into the ROI of the next cell, right? And when that dendrite, when that cell flashes, its dendrite that is overlapped into the other cell, to the neighboring cell, that creates a problem because if you have this ROI, you'll be contaminated with the signal from the cell next door. So that's why, yes, you can use these kind of algorithms that look at ROIs per se, but ideally, what people tend to be doing these days is not to look at the spatial features of cells, but look at the way that the signals themselves change, and look for pixels that change simultaneously. And you can do this. One of the first papers to do this was Eran Mukamel, when he's still with Schnitzer at Stanford. And he used this PCA and ICA-based algorithm to automatically segment cells. So basically, by looking at pixels that have the same variance, the principal components, variance-based, things that are increasing at the same time, and you say, well, there's all these pixels here that are increasing at the same time, so I'm going to call this a cell. Now, that is a problem, too, because imagine that you have cells that are highly correlated, right? Then if they're highly correlated, and so there's kind of, you would be detecting the same kind of areas for one and for more than one cell, because they're varying kind of, at times they're varying simultaneously. So that's why you create all the sorts of filters to go with this, and you create a PCA but also an ICA algorithm, and you can identify even spatially overlapping neurons, things that have ROIs that are complex. Like, for example, in this example of the initial paper where they published this method, they had Purkinje cells, which are very hard to track, right? Because it's very elongated. They're all aligned. They have these very large dendritic trees. So you get these very thin, long, elongated structures of their where you can see their activity. And so they first did this dimensionality reduction using PCA, and then they kind of separating the mix from different cells using ICA to extract, so independent component analysis, things that vary independently to extract individual signals. And this was a step in the right direction. Actually, a lot of people use this approach, and it works very, very nicely. So you actually solve this problem of not relying on the footprint of each cell, but you're actually looking at the signals and how these signals vary in time. What is the problem with here? Can you guys think of any issues with this approach? What happens if you have a cell that doesn't have any firing event?
Then you obviously can't tell if it's just background or not.
Right. So even if you have a cell that is clearly there, that you see it beautifully, structurally, but it had no event, no firing event, you really, you won't detect it, And the thing is that in reality too, this may not be too much of an issue, because if it doesn't fire, how can you be sure that it's a healthy cell? Or how can you be sure that it was labeled properly? Maybe it can be even bright enough for you to see it, but maybe it doesn't have the right concentration of the sensor and so forth. But that is an issue. You're only really looking at cells that are active at any given time. Now, so these tools, there's other tools that are similar, and one of the latest iterations and evolutions of this has been CNMF. CNMF is kind of a bit of a mouthful. It's called constrained non-negative matrix factorization. It's basically a method, a pipeline for doing denoising, deconvolution, the mixing of calcium data all at the same time. So constraints, meaning that it's called constraints. So the constraint part, meaning that you're trying to solve this. Yeah, Rowan, you have a question?
Yeah, about the last slide with cells not lighting up if they're not active. Would it also be appropriate to say that if the cell doesn't light up in the event you're recording, that it's most likely not involved with that event? Is that something that's like acceptable to say as well?
Absolutely. But there may be ideas, there may be situations when you want to look at counter-correlated activity, when you're actually interested in the silence. But again, for you to be able to say that, then you would need to positively verify with some sort of event, positively verify that this is actually a cell, and a cell that is firing, right? So
Could I make a comment on that?
Yeah.
So just to say, you know, so this, we talked a little bit about in my lectures about how electrophysiology has this huge sampling bias where you're recording only from the cells that are most active, and that it, for a long time, gave people these very false notions of how the brain works, because they think it's full of all these cells that fire at 30 hertz all the time and that sort of thing. And so there's also the danger of that sampling bias too, if you're only seeing, so it's sort of each, you know, so there's less sampling bias than this, but it's still a potential sampling bias in a place like the dentate, where activity is very sparse, and that's physiologically, we know that's how the dentate actually works. So anyway, just a comment.
Right. Yeah. No, that's a very, very, very good point. So CNMF, I'm not going to go into the specifics of CNMF. I'm going to say that, you know, this is basically solving an equation for two variables, and I'm going to talk to you about what those are. And you apply these constraints that, you know, that you have non-negative spatial and temporal components. So signals that are non-negative, meaning the activity goes up whenever, so there's higher fluorescence, it doesn't go in a negative direction, and you have non-negative spatial components, meaning your cells are actually positive pixels. I don't even know what that would be for a negative, I mean, it wouldn't make any sense. But these are the constraints in the non-negative part of matrix factorization. And matrix factorization is just a way to solve your equation. And you have a bunch of different constraints that you still use, like the cell size and the cell shape, the fact that your signals are sparse, and that you're expecting temporal activity that follows the calcium indicator dynamics. So you basically try to, you're trying to solve an equation with multiple unknowns, and you basically try to constrain your variable space so that you have as few as possible. So you make it easier to solve this equation. And I must say that I did something a little bit unconventionally here, because I'm totally kind of stealing. I didn't ask for his permission, but I will. That's why I need to acknowledge him very much. This guy, Pengcheng Zhao from Colombia, I think he's in a lot of Liam Paninsky. And I saw this YouTube video where he gave a talk at MIT talking about CNMFE. I think is a version for the microendoscopes or miniscopes. I think that he was very involved with, but I really liked his slides. So that's why I'm showing you like 4 screen captures of his slides, and they're not my slides. I again repeat, they're his slides, but I highly encourage you to have a look at his talk. So basically, whenever you have calcium imaging data, you have these time varying images. And they're really composed of different components, which are your neurons that have a time-varying fluorescent signal. And it's neurons 1 through K, plus a background, plus a noise. And each one of your neurons is basically, this spatial temporal activity is basically whatever your neuronal shape plus the actual temporal activity, which is what you're interested in, right? Your signal. So one way to display this would be that your data Y equals all of these different neuronal components plus B, background plus noise, and each Y, each neuron, it's really your neuronal shape, which is the footprint of your neuron. Is basically what shape it has on you know, whether it's round, whether elongated, whatever. Then convolved by this temporal activity, the actual signal. And this matrix factorization approach is basically you create these 2 matrices and you solve this equation of Y, Y being your data, equals AC, and A is the spatial shape of your neurons, C is the temporal activity of one neuron, plus the background, plus noise, which is E. Sorry, I'm flicking around like crazy, plus a background noise. And what you're going to be doing is solving the cell extraction problem, which can be solved by finding A, which is the shape of your neurons, and C, which is the temporal activity. And CNMF basically just solves this equation for A and C. And you have a bunch of different constraints that make it easier to solve this equation. And you have, you set, for example, what kind of cell types you're expecting, how big they are, what spatial shapes you're expecting. The more you can constrain it, the easier it becomes to solve. The kind of activity, you know that it's going to be G-camp, so it decays with a certain temporal decay constant and so forth. Nevertheless, whenever you do this, and this is an example of processing of data using Suite2P, which is one of the popular tools for doing this. Whenever you do this kind of processing, automatic processing, there is a lot of curation involved. You need to go in and look at your signals. You need to look at your cells. Do they look like neurons? Do the signals look like the signals of neurons? You can never completely rely just on whatever signal you get from your computer, because then stuff starts going wrong really fast, because you'll have cells which are maybe combined two cells, cells which are not cells at all, but they're background and so forth. And so for human supervision, there's GUIs available. This one, there's this little video that is playing here. It's a GIF, and it's from Suite2P by Marius Parchitario and Carson Stringer. We actually use this in the lab. And there's another one, another GUI from a tool called Calman by Eftichios Nevmatikakis. I don't know if I pronounced that right. But anyway, look at both. Those are right now two of the most popular ways of analyzing calcium imaging. So you basically, what these things give you is they give you the cells, the space, the footprints of the cells, the ROIs, and they give you these signals out. And okay, so now you manage to get the signals. How do you get there from those signals to your data? And I need to keep an eye on the time because I'm running out of time. But you know, after you get your signals, you may need to have a few other, whatever method you used, whether it was one of these automated methods where there's just an ROI that you drew with your hand. You may need to filter your signal. This would be example, e.g. With a Gaussian filter, for example, a window at 3 seconds, do some sort of filtering to remove noise. You want to also calculate your baseline fluorescence, because you're looking at signals that are varying in time. So your baseline is going to be the fundamental thing to figure out, okay, what kind of activity do I have? I need to compare it with the baseline. And a baseline is really a short pass filtering exercise. So you have whatever is fast varying, you keep, and you do, for example, one way of calculating the baseline is to do the median of the whole trace. Depends a lot on what kind of trace you have. Sometimes you do a mean of frames before stimulation and after stimulation, if you know that you're expecting a signal at a certain time, which is, for example, when you present the mouse with an odorant, or when you flick its whisker. And then also a mean or a median of a rolling window, in case you want to you just have a mouse running around and you just want to look at activity that is not constrained by a certain specified stimulus. So you look at this, you do this rolling window, for example, 15 seconds, and either the mean or the median will be your baseline. And then you calculate what's called the delta F over F. That is, you normalize your activity to the baseline. And then with this, you can do a variety of different types of analysis to that. You know, one way to do this, to analyze stuff is, for example, here, you have your delta F over F, and you have a stimulation. In this case, it was an optogenetic stimulation from that same paper that I showed you early in the orbitofrontal cortex, where they stimulate a few cells with optogenetics. And you get this baseline, and sure enough, when you stimulate, you have a certain increase in activity. And you see here in this raster plot, how the activity changes. And that could be as simple as that. So you calculate how much, on average, how much delta F over F you have. That's one type of analysis. Another type of analysis, for example, to detect any deviation from the baseline. You have a mouse running around. This is taken from the from one of the very famous initial mouse on the ball in the virtual reality papers. So these are recordings from CA1, from cells in CA1. And you have your transient and you have your baseline that you detect using a window and looking at the average of a rolling window. And then what you say is, well, I'm going to define anything that goes above 2 sigma, those 2 standard deviations from my baseline. I'm going to define that as an event. And these events, they're denoted here in red. So the red stuff is whenever you had a calcium event, a cell, each one of these 1 to 30 is a different cell. Whenever it goes above this baseline and you have a calcium transient, you detect it because the signal just basically went over 2 standard deviations of what it's supposed to be. And you see here, there's some cells that correlate very highly with epochs where the mouse is running. So this very bit, the very last trace is the movement of the mouse. And you see there's a lot of these neurons here at the very end, they're entirely correlated with the running of the mouse, right? So this is one form of calcium event detection. Now I'm just coming to the end. Another way of analyzing your data is to take a different approach and to say, well, we're actually interested in the firing of neurons. So the whole point of this exercise is to look at how the firing of neurons varies with time, and that's why we're doing calcium imaging. So is there a way where you can correlate electrophysiology with the calcium signals? Now remember, we've covered this a few times over this course, that your calcium signals are actually much, much slower than action potentials. But whatever calcium signals you have, they're going to be the deconvolution of action potentials. And here I'm showing you that this is a whole second of recording, and you see here the scale, and the action potentials are maybe like 1, 2 milliseconds, 3 milliseconds, whatever. And this is deconvolved with this response for a single action potential. I mean, you have several here that are plotted, but it doesn't matter really. Some are faster than others, but look at the time scale. This is half a second. So it's a much, much slower event than this. And you have this convolution. So what you can do is, and it was done, I think, for the first time by this paper in Yaxi and Rainer Friedrich in 2006. So they deconvolved is the deconvolution to estimate the firing rates. And they basically used only 2 parameters. One of them is the decay time of the fluorophore. That's the tau of this curve here. And also they applied some sort of threshold for noise, saying, well, if it's a very small transient, we're not going to analyze that. And then you deconvolve your signal. And this actually increases the temporal resolution of your calcium sensor, because by deconvolving, you actually get the temporal resolution, which is much, much smaller than the decay time, which is in the order of seconds of your calcium sensor, like your GCamp or your JR Gecko or whatever. And then the output of this is estimated firing rates, or in some cases for other algorithms, it's going to be action potential firing probabilities. And this is one example here. So this is data collected in I think it was Jake in my lab collecting this, that you have raw fluorescence and of C1 neurons, and you have these beautiful calcium transients, and you can get this deconvolved signal where these are arbitrary units and these are fluorescent counts. And then you always have a little bit of a baseline, and you can do further thresholding where you get pretty much something that with some fidelity reflects the kind of activity that you're seeing, which are these 3 large gradients and these 2 blips here at the end. And that's kind of like, right now, it's probably the most commonly used form of analysis of calcium transients. And again, a lot of things work, but it depends on what you actually want to do. So this is pretty much the end. We're at 12:22 now. It's pretty much the end. I just wanted to tell you that there's a bunch of resources online. I already mentioned a few of them, the papers, the videos. There's the Sweet 2P, CNMF, Norm Corey. They all have excellent documentation and their own GitHubs. There's videos online from Karsten Stringer and Marius Parcitario. There's a few blogs that are very useful. For example, Lab Rigor by Spencer Smith at UCSB. Fluorophore Spectra I showed you. And this blog actually has a simple explanation of CNMF. And these are the two photon spectra that I told you about. So how to look with it, how to check if a fluorophore is a good two photon fluorophore. And now I think I left about 5 minutes for questions that you may have.
I had a question about last lecture. You talked about deconvolving the point spread function.
Uh-huh.
Is that something that you usually do to get like higher spatial resolution, or?
Usually not with in vivo data, but you can with, and people do with data that is more static from confocal images and so forth. And in fact, a lot of, or I don't know if a lot, but some of the confocal software, the commercial confocal softwares, they actually do that for you automatically. So yeah, but that is a commonly used technique, but not so much for in vivo recordings, no. Other questions?
Just out of curiosity, is it possible to have like a mouse line with a fluorescent soma while you're doing in vivo recording, so you can identify cells without?
Oh yeah, that's an excellent question. And this is actually very useful for different things. You know, there's different ways of solving this problem, which is the fact that your cell may not be visible with GCamp, because GCamps were developed to have the maximum possible delta F over F, meaning the maximum possible transient. And one way that they did this was by decreasing to a minimum the baseline fluorescence. So these things are really, really dark unless the cell is active. And that can be a problem. For example, if you're trying to find ROIs, anything that is not based on a cell, detecting a cell that is active, that can be problematic. So yes, you can, there's two ways of solving that. One of them is that there is these GCamp 7B, for example, this is purple line here. So this is a GCamp 7B is a GCamp that has a higher baseline fluorescence. So this is really useful for looking at, for example, axons and smaller structures, because you need to know, okay, is this thing still there, or did it just go out of the plane of view, right? But you know, increasing the baseline fluorescence comes at the cost of your response size, right? So the fast version, which is the one optimized just for the highest possible transients, it's much bigger, so your responses are almost double the ones in 7B. But you're absolutely right. There's a bunch of viral vectors that actually express, say, a nuclear red dye or red fluorophore, like a T tomato nuclear, and then GCamp in the cytosol. So that really makes it easier to track the cells and to keep track of them, basically, and to know how many you have. But again, not having a transient from a cell doesn't mean that the cell is, so there's no way that you can tell that the cell that doesn't have for example, it never fired because it's unhealthy. You know, that could always be a possibility. So that's why it's sometimes hard to look at, you know, what is the proportion of cells responding to a certain stimulus? Well, yeah, you do have a good, relatively good estimate, but it's not without caveats.
Could I make also just a comment on that about the neurophysiology of that, which is that the notion of a cell responding to a stimulus or not is a very sort of artificial notion. So people have done studies where you look in like auditory cortex at the cells that quote respond to the stimulus versus the cells that don't. And something like 10 or 15% of the cells respond to the stimulus. But you can decode the stimulus identity better by looking at the cells that do not respond to the stimulus than you can by looking at the cells that respond to the stimulus, because there's more of them. So there's sort of a baked in assumption about cells having this big response to something versus not, when that's not really how the brain actually seems to work.
Yeah. So there's a couple of acknowledgements that I should do. Some of the stuff, some of the images that I've shown were acquired by people in my lab. So that was mostly Agustina, who had the code, wrote some of the code and acquired some of the images that I showed. Jake also, I think, acquired a couple of the images, and Kelsey may have acquired some of the spine data that I showed you. So it's just that some of the stuff from the lab was not acknowledged properly. Okay, that sounds like you guys are questions. Any more questions? I think we finished right on time. It's 12:29. Okay, that's it. Well, you guys stay safe. Enjoy your break. As I said, this Thursday there's this there is this lecture online, Optical Society of America, on that should be really good. She's a great speaker. I highly recommend it if it's a topic that you're interested in. So they're really going toward whole optical interrogation of circuits. So activating not only readout of activity, but also activating and inhibiting cells with light. Okay. You guys stay safe, stay healthy.